Hello, everyone. Thank you very much for attending today. My name is Claudia Labrador. I am the Global Product Manager at Stila. I will be your moderator for today's webinar session, The Rise of Digital PCR Applications, A Decade's Journey. I am pleased to present our speakers today. So I start with Dr. Jim Huggett. He's the Director of Biological Metrology at the National Measurement Laboratory, the NML, hosted at LGC. It's the UK's designated National Measurement Institute for Chemical and Bioanalytical Measurement. He received his PhD at Cardiff University before moving to UCL to work with professors Alimuddin Zumla on the molecular diagnostic of infectious diseases, focusing on low and middle income countries. At the NML, he continues this work on addressing the challenges of diagnostic standardization and test accuracy. He is a fellow of the Royal College of Physicians of Edinburgh and Royal Society for Tropical Medicine and Hygiene, and represents the UK on ISO and several key diagnostic committees. Dr. Huggett is also a visiting professor at the University of Surrey. We have also Dr. Alexandra Well. She is a science leader in nucleic acid research within the NML. She is involved in nucleic acid metrology projects requiring complex digital PCR assay design for the quantification and characterization of viral vectors, gene circuits, gene editing via CRISPR, and the detection of cancer biomarkers as well, including minority variant detection and copy number variation. The focus of this work is to identify and support the metrology framework required by developing standardized and higher order methods to enable then accurate characterization and production of high quality and reproducible end results. She has extensive practical experience using different commercial dPCR platforms and has given many presentations and conferences on experimental and analytical considerations needed for digital PCR. And then we also have Ward D. Spigelaere. Is an associate professor at Ghent University Faculty of Veterinary Medicine. He holds a master's degree in biology and a PhD in veterinary sciences. His current research primarily focuses on veterinary oncology and digital PCR. His work on digital PCR aims to enhance the precision and utility of this technology in various settings, from clinical to basic research applications. He's also a member of the MIKE Consortium and a spokesperson of the Ghent University Digital PCR Center. This center provides help and services on digital PCR and provides yearly courses and performs basic research, but also benchmarking studies for digital PCR. It develops novel data analysis pipelines for automatic digital PCR data analysis, including partition classification and downstream data analysis. So really, we have the experts of digital PCR here with us today to talk about the evolution of the last few years. I take the opportunity before we start the session to remind you that as part of our webinar series, Stila will be hosting as well a session on precision QC for gene therapy for monitoring of DNA concentration and purity with digital PCR that will happen May 22nd with Evan Anker from Canon Therapeutics at 10 a.m. Eastern Time or 4 p.m. Central European Time. So that being said, I wanted to also mention that we will have questions at the end, and you can submit those throughout the webinar session. So with that, please join me in welcoming our speakers today.
Good day, everybody. It's great to be here. My name is Jim Huggett. I'm the Director of Biological Metrology at the National Measurement Laboratory at LGC, and I'm also a visiting professor at the Faculty of Health and Medical Sciences at the University of Surrey. It is a great pleasure to be with you today, and I'm going to give you a brief introduction on digital PCR. Before I do, I've got this disclaimer I need to show, which basically explains the fact that we're going to talk about digital PCR instruments, we're going to talk about digital PCR, but we're not in any way endorsing any of the instruments. And just because we're showing the instruments doesn't mean we think it's better than one or another. I am, however, an advocate for the technology. I think digital PCR is a fantastic method, and so you can argue that I'm a bit biased when it comes to that. So let's talk about PCR. So PCR, the origins of PCR, some of you may be aware, was developed by Kary Mullis, working closely with a team at Cetus Corporation in California. And this was 1983. So PCR is over 40 years old now. And Kary Mullis also got the Nobel Prize for this. So it's not it's a pretty powerful technique, was well recognized. Early on, it was a fairly kind of time-consuming method where you would run a PCR, you'd set it up, you run temperature cycling, and you'd have to run a gel. to see the prize. And at the time, we're talking 35, 40 years ago, other methods were also developed to kind of compete with PCR, particularly isothermal methods that did not require the temperature cycling some of you may be familiar with when it comes to PCR. At the kind of turn of the millennia, 20 or so years ago, PCR moved and was developed into qPCR, a real-time PCR, where you combined the PCR reaction with a fluorimeter, or able to see the increase in amplification as a fluorescent signal when conducting PCR in real time. Then what we're talking about today is digital PCR and also a stream of high throughput PCR, which is particularly relevant in some scenarios such as in diagnostics. So this is kind of all variations on what we call nucleic acid amplification tests or technologies, or the NAATs or NATs. It's probably not putting too strong a word on it that actually the molecular methods, the NATs and especially PCR, have been absolutely instrumental in driving life science. In fact, if you think of a method that's been developed over the last 100 years, you'd struggle to think of one that is as powerful as PCR. I mean, there will be others, but PCR is absolutely up there as one of the most powerful techniques. If you think of its impact on life science, if you think of sequencing, the Sanger method for sequencing is essentially PCR. So it's an incredibly powerful technique, very versatile. And without PCR, we probably wouldn't be anywhere near as advanced as we are in life sciences and the medical space when it comes to bioanalysis without this particular method, whether it's genotyping, diagnostics, environmental monitoring, foods analysis. You think about it, it really is a key method. So PCR is generally, and NATs are generally really powerful and heavily involved in most life sciences. If we focus, however, on digital PCR, what are we talking about? So if we look at this schematic of a tube with some DNA molecules in it, which is how you might think of a qPCR reaction. And it's run in a bulk reaction, and you amplify and you increase the number of amplicon DNA sequences and quantify according to that way. Digital essentially takes this idea and just partitions it, meaning instead of having one 20 microliter reaction, in this example, you could have 20,000 1 nanoliter reactions, each containing a DNA molecule or not, depending on the concentration. And so if you zoom in on this, you can see that you have these individual partitions. Some have nothing and some have a DNA molecule. And this is essentially limiting dilution. And the PCR is performed as normal using standard real-time PCR chemistries. And depending on whether a DNA molecule is present or not, you will get a signal, an increase in fluorescence. And that is how we determine whether a DNA molecule is present. And then we can conduct absolute quantification by looking at the positive and negative partitions and apply Poisson statistics where multiple targets may occur per partition. Now, to do digital PCR is actually conducted pre-qPCR by hand using 96 or 384 well plates. That's incredibly cumbersome and resource intensive. And it's the development of commercial platforms over the last 20 or so years that has made digital PCR a going concern. And actually, the last decade, some really big developments in both in partitioning, whether that's emulsion partitioning or the droplets you may be familiar with, or solid phase chip-based instruments. These all allow you to do the partitioning, and it's the engineering around the partitioning that has essentially allowed us to do digital PCR in anger. And so we look at where we are today, and you see there are a number of newer instruments coming on, number of different providers, different ideas, different solutions. And to the extent of where we are at the moment, this slide is probably already out of date as new ideas, new instruments are coming on. The take-home message is digital PCR is here. It's a technology that has come of age, so to speak, and so it really is an exciting time to be talking about it. Why would you consider digital PCR? Well, it has superior accuracy, whether measuring minority genotypes or low copy molecule measurements. Both in these levels, it provides you with very good confidence and often does not require calibration curve because you're counting the molecules or you're counting the positive partitions. There's no immediate need for calibration curve in the way you might for qPCR. There may be situations where you need calibration, but generally it's much, much easier to perform. It's much easier for you and I to get a comparable result when measuring the same thing. It is also more precise than qPCR. The distribution of the molecules dictates the precision. So we can predict it very, very consistently, and it's By increasing the numbers and increasing the concentration and replicate measurements, you can increase the precision. And we've got down to 1.05% changes now, which is completely unprecedented with PCR type methods. And it provides you very confident reproducible quantification. And then the final point here is it provides unique analytical capabilities like linkage, which my colleague Ward will be speaking later on in this presentation. I'd like to focus briefly now on the high accuracy component. And this reflects a study that was conducted about a decade ago as part of the Nucleic Acid Analysis Working Group of the CCQM, where we looked at digital PCR and compared it to other SI traceable orthogonal methods. And you can see from this figure, there's very, very good agreement between different methods. Key here is the digital PCR is uncalibrated. And you can also get very good agreement between laboratories, again, across the world, measuring the same thing without calibration. And the difference here of 20% is, it's significant, you can see, but it's only because the method is so precise, and we are working to understand why there are discrepancies between that. But I tell you now, thinking of absolute quantification, there are no other methods that allow you to do this without calibration, with being so reproducible. This accuracy can be applied in the context of potential clinical scenarios like precision medicine, and this is a study published in 2018, where we demonstrated that we get very good agreement between different instruments. So it's not instrument dependent. If you're getting the partitioning right and the PCR works, you're going to get very, very good agreement. This very low signal, very accurate, very good agreement. And this represents one of the first examples of a reference measurement procedure for molecular methods on the Joint Committee for Traceability in Laboratory Medicine database, which is more the space of the chemistry, the clinical chemistry, where they have much more accuracy. It's a much more developed technologies involved in clinical chemistry. You can see here molecular methods are beginning to frequent the same space as the high accuracy clinical chemistry methods. And then this is just out of earlier this year, looking at using this method to measure pathogens, in this case SARS-CoV-2, the causative pathogen of COVID-19, with very high accuracy to underpin reference measurements for quality assessment. Also, examples of applications of digital PCR, just to point out, in high accuracy measurement of foods. In this example, genetically modified organisms, where the European Commission, JRC, are publishing guidelines on how to perform digital PCR in this context and applying it to support GMO testing to measure the presence of GMO in potential foods. This also refers to the digital MICA guidelines. And I will my final slide to you before I hand over to my colleagues is some additional reading. The digital MICA guidelines is the minimum information for publication of quantitative digital PCR experiments. And this was updated in 2020 to in line with the developments of the technology. And this is a really good document to go to for just kind of basic questions and answers you might have. And it also guides on how to publish the information that should be or ideally provided when you're publishing using digital PCR. And for those of you who may be interested in the accuracy component, high accuracy DNA and RNA measurement, I refer you to the manuscript below, which is talking about metrology frameworks, measurement science in the context of nucleic acid analysis and how we're progressing with that. So thank you very much. I will hand over to my colleagues now who will continue the session.
Thank you, Jim. Hi, I'm Alexandra. I work with Jim at the National Measurement Laboratory. I'm a science leader in nucleic acid innovation. Spend most of my time doing digital PCR and sequencing. And so today I'm going to talk about assay design for digital PCR and the exciting opportunities that this offers. So digital PCR has the unique partitioning that other PCR formats do not. It allows us to perform the absolute quantification by counting, as Jim has just talked about. But it also gives us a much more straightforward route to multiplexing. So when we think of multiplexing, we traditionally think of each target that we're interested in being detected with a different dye. So duplex would be the detection of 2 targets with 2 different dyes and 3 targets with 3 different dyes, as shown here. And so we can see the 8 different clusters and start to quantify as well as look at identity of our different molecules. And we can go up to 7 targets with 7 dyes, as is now possible with some of the commercial digital PCR instruments that Jim showed you. But with digital PCR, what we can also do is called higher order multiplexing. And this is where we can look at multiple targets with the same dye. And so what I've got on the right here is just an example where I've detected 4 targets with 2 different dyes. And I'm going to go into the kind of output and what this is in more detail in the following slides as I talk about the three main types of assay design during this webinar. So to start with non-competing assays, this is very much traditionally what we think of as assay design. You design a primer and a probe for one target and a separate primer set and probe for another target. And your output of digital PCR is in the form of a one color scatter plot. So Jim showed you a few before, and I'm just going to talk you through this one in detail. So what I've got here is along the x-axis is 5 different unknown samples, 2 controls and a water. And then up that y-axis, I've got the endpoint fluorescence of each of the partitions that's shown as a tiny dot in each reaction. So this is a QIAcuity scatter plot. So there's about 8,500 partitions per reaction. Partitions with a higher endpoint fluorescence are above that horizontal line and are classified as positive, and in this case, colored blue. And partitions with a lower endpoint fluorescence are below this horizontal line and are classified as negative, and so colored gray. The red horizontal line is called the threshold, and it's positioned in that space between the positive and negatives, and should be universal across all reactions in the experiment. So we need clear separation between those positives and those negatives. So this is an example where we looked at the number of integrated lentiviral targets in human cells, and we can see that some of those reactions have positive partitions in them and some of them do not. But what we can also do is look at the scatter plot from the second target. So these reactions are duplexed, and here I've got a human target on a different probe, in this case a hex. And we can then look at the number of positive partitions for each of those 2 targets. So that's the number of partitions above the line, and apply lambda and the Poisson correction. So this is where we look at the proportion of the positive partitions, that is K, out of the total number. So I hope looking at this equation, you understand why it's important to be able to accurately classify which partition is positive for a target and which is negative. Because if we get that proportion incorrectly, we will end up with a biased or incorrect value. So I said this was a duplex reaction. So what we have with digital PCRs, we can also look at what's called a 2-color plot. So when I do an assay design, I tend to draw out what I think I might get in my reaction and then try and work out what kind of signal I will get. And so I'm going to just talk you through this. This is the same lentiviral example I gave you before, with an LV target as 1 with a FAM probe and a human target as target 2 with a hex probe. So a partition here is just shown as a circle. This is not limited to droplets. It's purely schematic. So these can be chambers as well. I could make these squares, but I'm just designating them as circles for this presentation. And so to start with, we have our empty partitions. They have no molecule in them. And then we have partitions that are positive for target 1. We've got the partitions positive for target 2. So that's shown with a blue or a green line here. And then we've got them positive for both. And then you can look at the endpoint reading and start to work out where these partitions will sit in this 2-color plot. So a 2-color plot here, I've got the FAM or target 1 up the y-axis and the hex along the x-axis. And so what we get is this rectangular configuration where we end up with the double positives that are just colored in orange here that sit with both a FAM and a hex probe. So in real life, this is what it looks like. So this is the QIAcuity. The colors don't match here. I apologize for that. Each different instrument has its own software. It colors them in a different way. So the yellow partitions here are positive for the FAM. So that's colored blue in the schematic. The light blue ones are positive for hex. That's colored green in the schematic. And then the dark blue ones are the double positive. And so what we can see with 2 examples here is this rectangular arrangement in the middle one that shows us that there is LV detected within these as well as the human. And so we can look at the proportion there and say that there is 1 LV copy per cell. That is, there's about half the number of LV targets as there are human targets, and these are diploid cells. And then the one on the right shows us that we have no LV integrated in this cell because we just get the human targets. And this is a hugely exciting study, literally published in the last week, demonstrating the utility of reference materials. It's headed by NIST, who then sent them out to 12 different labs. And our lab looked at them with real-time PCR and digital, but other labs use molecular combing. We've got NGS, and it's a hugely exciting resource and development of a reference material for LV integration. So hopefully you can see that this can be expanded up beyond 2 colours. And so here I've got an example of 3 targets to investigate drug resistance in bacteria using the Spigelaere system. We have different clinical samples containing unknown numbers of methicillin-resistant or sensitive Staphylococcus species. And from this type of assay design, we can get the pathogen load from the absolute quantification of the species-specific target, but we can also see which ones are methicillin-resistant and so treat accordingly. So the second type of assay design covers competing assays. The competing here refers to the probe, where each targets the same region of the amplicon using a single pair of primers. This assay design really took off about 10 years ago in the minority targeting space, where the partitioning of digital PCR enables the detection of mutations at less than 1% fraction abundance, so huge potential in cancer diagnostics. But importantly, this approach requires you to know the sequence of both your wild type and your mutant sequences. So here we've got a wild type probe on a hex, and we've got a G12D probe on a FAM. And there are 4 different molecules that go into 4 different partitions with the different outcomes. And so if we look at that 2-color plot, we would initially predict that we'd see that rectangular configuration that we saw with the non-competing assays. However, in this assay design, we have a single pair of primers to detect 2 targets. And during amplification, the reaction becomes limited. And we call this partition-specific competition. And it results in a drop in the end point of the fluorescence in both probe channels. And then that double positive cluster also expands out to form this arc between the 2 single positive clusters. This isn't necessarily a problem, but if we look at it in a real life example here that I've got with the QX200, we can see that that arc has formed between the mutant and the wild type only clusters, and we've ended up with this triangular configuration. This is not a suboptimal assay, but the result of the partition-specific competition. So while this may appear to be tricky, the challenge is really only to set that threshold to separate out the 4 different clusters. And we have seen that some assays are less effective than others, but if there is a single nucleotide difference between the 2 probes and there's only 1 primer pair, then this is representative of what you see. So the example here is the KRAS G12D assay that was used in the big reproducibility study using 21 labs, and I've given the citation down in the bottom right corner. And here we can use the same lambda equation to quantify the wild type and the G12D molecules. And this example, we get just under 30% of G12D in the sample. As part of this study, we sent 4 unknown blinded samples to the different participants. They had positive and negative controls. And it also describes the partition-specific competition in more detail. So this shows how you could use the controls to look at things like the false positive rate, but also how to set the thresholds, so the vertical and horizontal pink lines, to accurately classify the clusters. So the relatively new assay design, first described about 10 years ago, but it's really been in the last sort of 5 years when it's really taken off and you suddenly start seeing the number of publications going up. So this is a hybrid or drop-off assay. And this is for when you know there's a change to the target, but you either don't know what it is, or maybe you don't need to know exactly what it is, only that it isn't wild type. So either way, you can't design a probe specifically for the mutation or the change. If we think of partitioning those molecules, we have the same 4 outcomes. We've got our negatives, our unknown mutation, our wild type, and then that double positives. And so the name of this assay comes from the clever probe design here. So you have a single pair of primers. You then have a universal probe which binds to the region of the amplicon that you are not expecting to change. So this will count the total number of molecules that you have in your reaction for that particular target. So in this example, I've called it the universal probe and I've colored it green. So it's a hex probe. Then you design your wild type probe. So this is against the sequence you know. And this is blue, so it's got a FAM probe. And the assay design is called hybrid because those 2 probes are going to bind in tandem. The assay is also called a drop-off because for your mutant sequence, your wild type probe will not bind, but your universal will. So your wild type has dropped off and no longer gives you signal. So let's have a think about where those might sit in that two-color plot. So I've got the wild type up the y-axis and the universal along the bottom. So remember, we don't have anything specifically for the mutant. So our negatives sit within the normal space just here. And then our mutant will sit here. So we've just got the signal from that hex probe. And our wild type probes, our wild type partitions will sit here because we've got a signal from both the universal and the wild type. So it'll sit in the space that normally holds a double positive partition space, which of course creates a potential issue for partition classification. Because if you remember, I said right at the beginning, if we cannot classify our partitions correctly, then we will put the wrong numbers into that lambda equation. So we'll end up with a bias or wrong answer. So how on earth do we work out which ones are wild type and which ones are just a double positive? But on top of this, we have another consideration in that we've also got competitive PCR going on because we've got that single primer pair. And so therefore we get the expansion of the cluster, but we also get that drop in fluorescence. We don't drop in like we did for the triangle, we just drop down. So an experimental outcome to help you visualize this more. So this is a CRISPR experiment. So following Cas9 cut in the cell machinery repaired the error repaired the cut with non-homologous end joining repair. And in a number of cases, just doesn't do it quite right. And so we no longer have a wild type molecule. We're repaired as something else. So some kind of insertion, deletion, or maybe a nucleotide change. So in this particular example, we can't design a mutant probe because every time we have the NHEJ repair, it might do it slightly differently. So how do we quantify when we can't count the number of partitions that are positive for the knockout molecules, which is essentially what we're interested in? Well, we can use the same equation that I gave you before, but we need to rearrange it a bit. Before we were looking at the proportion of the positive partitions, but the equation actually stated that we do 1 minus the proportion of positives out of the total, which is essentially the same as doing the proportion of the negatives. So this means we can substitute in W instead of K over N and rearrange the equation. So we now no longer need to know the total number of positive partitions for our target. Feels really counterintuitive, I know, but I promise you this works. And so what we can then do is accept that we only need to put in these thresholds, these pink lines. We only need to classify these 3 possibilities, and then we can plug those numbers into the equation like I've shown you here to get the proportion. In this case, about 12% of the molecules are knockout. Now, I've gone through that very quickly. So this was originally all explained in this paper from 2016. So have a look at that, see what you think. And it's got a lot more behind the maths and shows you how you delineate from the original equation through to this second equation. So now for the fun stuff. So with these 3 different assay design types, you can actually start to sort of combine them in order to get even more information out. So I'm going to give you one example, CRISPR knock-in experiment. So here I've got the line on the left is my DNA molecule. The cut site is the predicted cut site for with the Cas9. And the knock-in will be possible by exploiting the cell's homology-directed repair pathway. So as before, with a knockout experiment, you're going to end up with a mixture of molecules. And I don't know exactly what the sequence of all of those molecules is going to be. So I need to designate them as some sort of knockout. And so I've called the molecule with a knockout, and I don't know what it is, as just X. We're then also going to have wild type molecules that either never got cut or were repaired perfectly back to the wild type sequence. And then we've got the knock-in molecule that has that extra bit of sequence that I've knocked in using the homology-directed repair, and that's the one I want. And then I've just got one example on the end here of a partition that contains 2 different types of molecules, but obviously there's 3 options here. So when we go about designing the assay, what do we know? Okay, so if I design a single pair of primers and a universal probe, then I've got a measurement of the total number of molecules in the reaction. I can design a wild-type probe that will only bind the wild-type molecule and use the same fluorophore here. So I'm going to put the universal and the wild-type both on green. I know my knock-in sequence because that's what I want, and that's what I'm really interested in evaluating, so I can design a probe and put that on FAM. Excuse me, my computer has just told me I need to charge it. Let me just do that quickly. Okay. So at this point, we're then going to have an amplicon generated that's either going to bind the universal and the wild type, or the universal and the knock-in, or just the universal. So we've combined in that idea of the hybrid drop-off assay along with the competing assay. So let's have a look at the endpoint fluorescence of the partitions. So I've got my two color plots here. I've got my knock-in amplitude, so the FAM up the y-axis. I've then got my hex for the universal and the wild type along that x-axis. And let's have a look at where our partitions might sit. So our negatives, dead easy. They sit exactly where we're always expecting them to sit. Negative for all three probes. Okay. So our knockout is then going to sit here because this is going to be positive for the universal probe only. And our wild type is going to sit here. So what we have is two hex probes binding in tandem. They're not competing. They will both bind on that wild type molecule. And so it gives you a doubling of the fluorescence in the hex channel. Then our knock-in partitions are going to sit here because they're going to be positive for the FAM, the knock-in, and they're going to be positive for the universal. So they will sit in this orange space here. But again, with this particular one, we're going to have that issue with the combined cluster that will sit in the same space and the competitive PCR, so we get the expansion. And we can't separate those two clusters. So we've also then got three other combinations of double and triple and positives that may occur. And I'm going to talk you through a couple of ideas that we have on how to work around that. So a real experiment. So this is where we've knocked in a flag tag in frame with the gene. So we can design a knock-in probe specific to the flag sequence and the wild type that covers the region where we have that predicted cut site from the Cas9. I've got this up as a heat map because it's actually slightly easier to understand than having the different pseudo colors that color in. And we've got different multiple thresholds to separate out, so we can count the number of partitions in each of those different clusters. And our knock-in is actually really straightforward because we know that everything that is above that horizontal line that's the threshold for the FAM must contain a knock-in, regardless of whether it's double positive, single positive. So our lambda for the knock-in is pretty straightforward to determine. The knockout and the wild type are quantified using the same equations from the drop-off approach. So I pop those here. And so using a combination of these, we can actually then calculate the proportions of the different molecules. And here we've got 5% knock-in, 27% knockout, and the remainder are wild type. So the goal here is to design an assay that starts to look at the efficiency of the CRISPR. So after this point, normally you pick single cells. And so here, by seeing that 5% of our knock-in molecules, you'd say, well, maybe I just need to pick a 96-well plate of single cells. That will be sufficient. But if we had, say, 1%, we might consider more. So I ran a number of controls here, including a wild-type only sample, looks at the false positive rate of the knock-in probe. We shouldn't have any in the wild type, but it also looks at the false negative rate of the wild type probe. It should always bind, it shouldn't drop off. And then I've also run quite a diluted sample here because what I found was having it too concentrated, you ended up with those extra 3 clusters that haven't really materialized in my sample there. And they were really diffuse and really expansive. It got incredibly challenging to understand how to set the thresholds. So this type of assay does have quite a narrow dynamic range. And then this is my final slide, serves as a summary of what I've talked about, the applications that I've mentioned along the way. We can use different assay designs to look at a huge number of different applications within digital PCR, and we can tailor those designs depending on the molecules we expect. It's hugely versatile, very bespoke. I've just focused on assay design in this webinar. I've had light touch on ideas for control samples, but each assay design needs optimization, as you would expect. On the whole, optimization process is pretty straightforward. The DMI key guidelines from 2020 touches on a lot of ideas on what you can do for optimization. In addition to this list, we're using advanced assay design to validate viral vector production. Our early studies involve putting viral particles actually into partitions and then seeing what targets are present to have an idea of what, how full the capsids are. A critical quality attribute of viral vectors is the identity as well as the titer. So we think digital PCR can be used to support both of these areas. And then another area I'm interested in is using digital PCR to look at the denominator of the metric. So a lot of the time we talk about copies per something. And one area I'm quite interested in this space is transfection efficiency. That is to say, how many vector copies have I got per cell that's been transfected? At the moment, there's still quite a big assumption that every time we do gene transfer using whatever method into whichever cell we're interested in, we get the same result. And I'm not sure we can assume this. And so I really want to take this forward, particularly looking in the massively expanding engineering biology space. And then I really want to finish by acknowledging the digital PCR experts in my lab, Dilek, Anna, Eloise, and Gerwin. Brilliant data that they've generated over the years that have allowed me to put together this presentation. And I'd like to say thank you.
Thank you for giving me the floor to present some of our research today. Before I start, I would like to quickly introduce our consortium. So I'm part of the Ghent University Digital PCR Consortium, and we have already over a decade of experience with digital PCR. We have been using digital PCR for multiple applications, from infectious diseases to oncology to mitochondrial chimerisms and so on. We are working across different faculties. Much of our research goes to digital PCR applications, but also to making new data analysis pipelines, such as partition classification or thresholding, and developing statistical data analysis tools for digital PCR. And we also offer training or help with designing your digital PCR experiments. And we also provide digital PCR as a service. So today I want to show you some of the work that we have done to measure intactness with digital PCR. And an example here would be if we make a gene construct with a gene of interest that we insert in a vector, then of course we want to know whether this DNA is intact. And with digital PCR, since we look at single molecule resolution, we can actually design different assays. For example, here a blue assay on the promoter region or in the promoter sequence, and a green assay in the gene of interest sequence. And then if we do digital PCR in a situation where all the targets are intact, and so where both the promoter and the gene of interest are linked to each other, then we would expect here in this dual color plot a cluster with no target. So these are partitions where no target is in. And then the intact targets should all be double positive for both the blue and the green channel. And of course, in a situation where there are some breakage, so some targets that are not intact, then we expect broken fragments, such fragments with the promoter only or with the gene of interest only in the only blue or only green channel. And the double positives, then the question is, what is happening here? We would expect the intact targets, but the story is a bit more difficult than that. And I will explain that showing you two situations. So this is a situation at a low concentration of targets. And if we do digital PCR, we would partition and the targets would fall into some of these partitions by chance. And at low concentration, we usually only have one molecule per partition. So in that case, we can say, okay, the only blue or the only green, they have broken partitions. And then the cluster, which is is both positive for the blue and the green, these are all the intact fragments. But now let's go to a high concentration of targets. In this situation, we have co-encapsulation. So sometimes we have partitions with only one molecule, Yeah? And in other cases, and those are more disturbing, we have co-encapsulation of different targets. And these can be intact targets, having always the double color. But sometimes we can have a combination of intact with some broken targets, or maybe even two broken targets of a different color. So the question then becomes, what is here? How many intact fragments do I have? Yeah? And this was the work of two former PhD students of our group, David and Yao, and they actually designed some or made some mathematical derivations, because if we know the broken fragments, so if we know the chance that partition will be positive for broken promoter only or broken gene of interest only, then we can actually derive from that the probability that these fragments will be co-encapsulated. And if we know the probability that these fragments will be co-encapsulated, we can actually know that the surplus of positive partitions is actually having intact fragments. And from those derivations, I will not go deep into the mathematics of this. You can definitely read the paper for that, but we derived this new equation where we actually look at the integrity of the DNA by deriving the intact fragments here. And we experimentally validated this mathematical formula by looking at different experiments with different concentrations. And here we have a low concentration scenario where in red you see the naive method where we would assume that all the double positives have intact fragments. And then in blue we have the situation where we use the probability statistics designed by David and Yao. And in black is just the expected value. As you can see at the low concentration, there was probably some pipetting error in our standard curve because we are a little bit off, but you see that the naive and the probability statistic method, they nicely follow each other. If you go to a higher copy number, so higher concentration with a lambda of almost 0.1, there we see that the probability statistics, there is some bias between the expected value and the amount of the concentrations that we measured, but we see actually that the trend really aligns nicely with the expected curve, whereas the naive method is off. And this is even worse in the situation where we have 30,000 copies. There, the naive method cannot be used at all, whereas the probability statistics still nicely aligns with the theoretical expected value. So this shows that using probability statistics, we can actually take out the ambiguity of these double positive partitions. Now, this was for two colors. We can actually also do that for multiple colors. And Yao made derivations for a four-color combination, where we have here one big molecule and four different assays, A, B, C, and D, to check the intactness. And here we see the intactness over time when we sonicate the DNA, so you expect some shearing, and we see that we do indeed see shearing of the DNA. The longer it becomes sonicated, we see indeed that it disintegrates. If we then also evaluate the intactness with the distance of the two or the different amplicons from each other, and we take sonication time in perspective, we see that the smaller fragments, of course, remain intact for a longer time and they are more robust. But the longer fragments, they start to disintegrate, and of course, this is also seen. This effect aggravates with time. Yeah. If you want to use this method yourself, you can design 4 different assays, and Yao made a digital Shiny application, which is freely accessible through this URL, and you can actually put in your own numbers of single positives for the different and double positives for the different targets that you are looking at, and then you will get intactness measurements with also a theoretical confidence intervals that has been or that is made with this estimate, because of course an estimate should always come with a confidence interval. Good. The other part of research that we have done that I want to show to you today is using this intactness method in the so-called CROCO study, and this is work from the postdoc and current core facility manager for Digital PCR Consortium, Wim Tripsteen, and he actually looked at the HIV genome, and he wanted to look how intact is the HIV genome inside infected patients. And for that, he designed an assay with 5 different targets throughout the HIV genome. And one of the studies that he did was actually just compare how these targets work across different platforms. So we have at our disposition 7 different digital PCR platforms. And we compare this. We don't have the data for the LOA and for the SNIPER yet. It's ongoing. The LOA data has been run, should still be analyzed, and the SNIPER data or the SNIPER experiments are being run as we speak. So this is quickly the experimental setup. Of course, not all the instruments allow an equal high number of multiplex reactions. And also for the BioRad system, we had it on loan and we were not able to get the 5th color working at that time. But all in all, we tried to use the same fluorophores as much as possible. We pre-allocated all the primer probe mixes to have the same amount of freeze and tau cycles over all the different replicates. We used as much as possible the same cycling protocols. All in all, we had 3 independently diluted standard curves for each of the systems, and each time we did triplicates. So for each different standard curve. Here as well, we tried to conserve the same freeze and thaw cycles, and in every system, we added 2 microliters of input material. And these are the dilutions that we have run. So here we first analyzed the data across all platforms just to show you how powerful digital PCR in general is. And these are the expected values of our dilution series. And this is what we actually measured. So the mean of all these numbers. And if you look at the coefficient of variation, that is also very, very narrow. So showing that digital PCR in a whole is very accurate and precise. So then we looked at the precision, comparing the different platforms, and these here are still the different dilution points. And here, every time you see these dilution points per system that we have right now investigated. So the absolute Q, the DLC from Roche, the NIO from Stilla, the QIAcuity from QIAGEN, and the BioRad system. And if you look across the different platforms and we compare them, we see that actually for all the different systems at the different dilutions, we have very comparable coefficients of variations. So showing that actually digital PCR across different platforms is quite repeatable. You might note here that the NIO had a higher variation compared to the other system, but here I think this is more a technical problem of some outlying dilution points, probably by pipetting error, because if you look the dilution points further off, they are very much in line with the other systems. So with this, I want to end my presentation. For the people who are interested in learning more about digital PCR, I just want to inform you that we are organizing a digital PCR symposium and a workshop in November of this year. Stilla is also one of the platinum sponsors of the symposium, so you're all free to register to this event. And if you have any questions, I will be happy to answer them.
Thank you very much, Ward, for the interesting talk on the probability statistics and the WAC through integrity calculations. I also want to thank Jim and Alexandra for their insights on the evolution of digital PCR over the last years. As we've seen, digital PCR has undergone remarkable evolution, and throughout this journey, Spigelaere Technologies has consistently stood at the forefront, transforming innovation into real-world impact. Just before we jump into the questions, let me introduce our latest system, the NIO and its features. NIO Digital PCR is an all-in-one system with a simple workflow. We just need to pipette the reaction mix into the chip consumables. These steps can be fully automated with standard liquid handlers, but then transfer these chips into the instrument where the sample gets partitioned before the PCR. At the end of the cycles, each fluorescent signal is read to extract the results in copies per microliter. The NIO counts with 7 colors for enhanced multiplexing capabilities. Results are obtained in as little as 2.5 hours, with the results being able to be analyzed in our analysis software, directly in your computer, or in the NIO with that enhanced flexibility. I invite you to check out our website to learn more with the QR code on the top corner, and listen to the testimonial of our users about how intuitive NIO is to use and why it becomes a game changer in every lab where it's implemented. Without further ado, let's open the floor to some questions. Apologies. Let's open now the floor for some questions. So I'm going to be starting on with some general questions about digital PCR. So let's go ahead and those. So I see a lot of questions coming in. That's very exciting. So starting with a little bit on the concepts on accuracy that we were mentioning before. How does digital PCR improve the accuracy of the quantification compared to qPCR, especially in low abundance or rare variant detection scenarios?
Do you want me to have a pop at that, Claudia?
I mean, sure, I can take the question.
No, I can have a go if you want. So I wasn't sure you wanted to go first. I'll pop it. Okay.
Yeah.
So the accuracy argument is often it's several layered, because a qPCR can detect low level molecules. In fact, if the qPCR volume is bigger, you could argue that the digital PCR is less accurate. But in the context of accuracy, as in not only can you detect it, but is what you're detecting correct, we've done a lot of work with the Metrology Institutes to show we have good confidence that when you measure 100 molecules, that is 100 molecules in a certain consideration. This is also done comparing with orthogonal methods like isotope dilution mass spectrometry, which is traceable to mass, and getting very good agreement from that perspective. And so we're confident that we get very good measurement of the number of DNA molecules, for example. There's also the fact that digital PCR cannot easily overestimate, because you can't get more than the number of DNA molecules that are present unless you separate the molecules before you do the partitioning. But even then, it'll only double. Whereas qPCR is in log space and can have huge discrepancies between laboratories. So it's much easier to get the agreement, and that agreement tends to be quite accurate. For the low rare trace detection or minority variant, well, we know that the qPCR it's linked to the false positive signal. It's linked to the fact that a wild type only template, if you're measuring, say, cancer variant or non-invasive prenatal testing, or choose, a single variant that Alex talked about, it's linked to the wild type giving you a signal, and that can be done with qPCR. It can cause probe incorrect binding, causing a pivot to the curve, and so you really don't have good sensitivity. I'll defer to Alex and Ward as to what that actually is, but it's not very good. With digital PCR, it's only really driven by the polymerase putting in the wrong or the wild type sequence and suddenly giving you that signal. And so it is much, much better at measuring that rarer variant than qPCR is. So that's kind of in the context. And so in that case, it's also more accurate, as in it's giving you what we consider a better or more accurate result of the right thing.
Perfect. Thank you so much, Jim. I actually am sorting some of the questions by subjects. We have one as well that actually says, bring on talks. Thank you all. My question is, how does digital PCR perform with environmental samples, especially ones with high amounts of PCR inhibitors like river water? Yes, I'm dispatching the questions, but feel free to jump in.
So I'll have a go at that one then, and I can stop speaking now. Given that neither, well, I know Alex doesn't measure on eDNA, and I don't know whether Ward has, you can pipe in. We are increasingly doing work on the environmental DNA topic, and digital PCR has become the method, certainly post-COVID-19. It was the method that people started using. Why? There's a lot of debate. Yeah, possibly the inhibitor component, although digital PCR provides you with more protection against inhibitors, but it's really important to realize it doesn't. It is, PCR is susceptible to inhibitors. Digital PCR is PCR, so it can, something that will kill a reaction will kill both reactions, qPCR and digital PCR. It's just digital PCR is slightly less affected by more subtle inhibitors that may cause what you might think of as a right shift in your qPCR. Well, digital PCR doesn't care. That'll be a rain. It'll be a lower amount as long as it's working. And so it's much more forgiving from that respect. The other reason I think digital PCR has provided more, has become more established with environmental monitoring, partly because people started using it and there is an air of fashion. But I think also that digital output, if you're measuring trace amounts of DNA, it's much more confident. You're much more confident you're getting a signal. My previous presentation, I said, qPCR can detect single molecules, but you're much more confident that digital PCR has detected the right thing when it does detect near single molecules, because that's what it does for a living, right? If you're measuring 10,000 partitions 10,000 DNA molecules or 10, the partitions are generally having very small numbers of molecules, 1, 2, 3s in them. So it has to work with that exquisite single molecule detection capability as a going concern. So, and therefore that I think is one of the reasons why it is potentially favored in the environmental DNA space as well.
Thank you so much, Tim. So actually now I'll switch over to assay designs, maybe some questions for Alexandra. So we have one question about that some targets sometimes, instead of going into one cloud of droplets, they get a trail of signal spread. If there's any, why could this be happening? How to avoid it?
So very good question, and also means looking at the two-color plot, which is excellent, rather than just taking the software's automated analysis as the true result. So it's slightly anecdotal at the moment. We haven't done any experiments specifically to this, but what we do observe is the more targets that are being amplified, that is different targets. So just to be clear, if you were doing a 4-plex, partitions that are positive for 4 targets tend to sit at a lower endpoint fluorescence than ones with 3 and with 2 and with 1. And so we are starting to see that complication in the plot. So you don't get the nice rectangular configuration that you might be expecting. You start to sort of see multiple clusters within each of the quadrants. You obviously have the ability to look at the 3-dimensional, the 3-color plots with the Stilla software, but once you get up to 4 colors, you then need to look at multiple 3-color plots. So it helps on one order, but then we need to start developing ways to analyze and understand software, humans understanding what we're looking at. And so we've observed it, we see it happens. We think it's because we're asking a huge amount of that tiny partition to amplify up 4 different amplicons. Probably a resource issue. At a certain number of cycles, the tack's tired. You probably haven't run out of primers and DNTPs, but we know that in each of those partitions, you get the little sigmoidal curve. We just only see it when it's at its plateau stage. So probably tweaking with parameters. I know that different manufacturers produce specific master mixes for multiplexing with higher target numbers. They probably contain a bit more reagents and other considerations in order to try and help those hard-working partitions that are amplifying more than one target. I hope that answers the question.
I think it's very interesting. That's a very good link. There's another question about complex, besides these complex cases, when we want to take into consideration some particular features when it comes to primer prep design specific to digital PCR, how the design could be different from regular qPCR platforms, for example.
So the design isn't different. So if you're doing non-competing, it's exactly the same. If you're doing competing assays, it's similar. Real-time PCR just gets in a bit of difficulty once you get to sort of 5% minority. And that's really just the mismatch. Jim talked about that in the first question. Hybrid or drop-off assays do not work in real time. I've had quite a few goes at trying to get it to work, but actually what you're trying to use a sigmoidal curve to interpret when something doesn't happen. And real-time PCR, it has to be efficient. You need it to be working the best it can, so close to doubling. If it's not doubling in each cycle, you need to know that, so you can then incorporate that into your interpretation of the CQ values. So the assay design in terms of non-competing assays, pretty much the same. When we started off, we used to run our assays on real time, transfer them to digital. We no longer do that. We just go straight into digital because we find digital PCR is pretty robust. If you run an annealing temperature gradient, if you run a primer probe titration, essentially you can get assays that work at from like 52 up to 60, maybe not quite as far, yeah, about 64 degrees for an annealing temperature. You get beautiful separation. Other assays, you don't. But it's certainly much more robust in that capacity than real time. And so if you go with what real time says is the best optimal cycling conditions, you're maybe restricting the options available to you for digital.
Can
Thank
I
you
add
so
to
much.
that? Yeah.
Yeah, so actually we're already on time. We do have a lot of questions. I did wanted to have a question on the integrity part with Ward on the relevance of actually doing integrity analysis with more than 4 or 5 targets when we're talking about these multiplexing assays. And then if you have time, we can stay a little bit longer with the questions. If now I can always transfer the questions so we can address with those offline.
Okay, so your question is whether what is the added value of having more than 2 targets for integrity? So I think if it's just to know the quality of the DNA, and if you're just interested in to have a certain length, then having these extra targets is not really interesting. But of course, you might want to optimize your assay for DNA extraction or whatever. And in that case, it's good to know where along the line you are, how bad is your isolation method. So for optimization purpose, it's good to have more subtle knowledge of what's happening. But then, of course, if you're working in viral vectors, you're making a new viral vector, you also want to know whether each of your constructs is in the vector, or whether some of the or part of your construct is lacking part of the genes of interest, for example. So you want to know the integrity. Do all the parts fit in viral vectors? And then the third reason, and this is more a niche reason, but this is actually why we developed it, is that we were looking at HIV. It's actually so Wim Tripstein who developed it, and he was looking at HIV, which integrates in the genome. And one of the big questions is how much of this HIV is still intact, how much of this virus is going to be able to replicate. And then we need to know we need to know all the components within this virus. It's not going to give us information of small insertions, deletions, but the bigger gene the bigger gene deletions can be found with good assay design.
Thank you so much, Ward. So maybe I can have like one more question, and then we can close up the session today. There's a lot of unanswered questions, so we'll get to those via email. So we have one person asking about the instruments that have 5-7 channels and what will be the number of maximum targets that you can detect in a single well if you use radial multiplexing, having multiple probes for a single target at different concentrations. And if one of you have actually tested these before.
I think the limit will not be the colors. I think the limit will be the interference of all the different primers and probes in there. That would be my answer here. And I wouldn't know the answer on how many. I think it's also going to be assay dependent.
And the software's ability to actually be able to analyze that. Alex is probably, I'm going to put Alex on the spot because you've had a go at this.
Yeah, so I've done quite a few combinations. What I have noticed is, I mean, there's papers now where they're designing assays that have like, I think it's 22 targets might be the highest. But one of the things that is quite clear to me is there's 2 things going on. So as far as I know, the most positive targets detected in every sample is around the 7-8 mark, possibly up to 10. I'm just suddenly remembering another paper, but what tends to happen is when you're multiplexing at the kind of 20-something number, you're not expecting them to all be positive. So it's this really cool way of doing like I think, you know, things like a respiratory panel. So there's like 18 different pathogens in there. You're not expecting all 18 to be positive. You're expecting 1, possibly 2. And so although it's
Really
like
ill.
it's really ill, yeah. I mean, then we're in trouble. But it's so you've got this high level of multiplexing where just the presence of the primers needs to not interfere with the ability for the target to be amplified that's actually in there. But the viral vector work that Ward just talked about, and especially with the HIV, that's really interesting because you are hoping for the positive result for all of your targets to be present. And that's a really, I'm quite excited about all the experiments that we can do in the next few years, just to try and work out how we can optimize that, how we can make it the best it can be. So yeah, I was going to say it's our imagination that is the limit on how much we could add, but I think it will be software without a doubt, and that the primer interference.
And to remember,
you
there's
so much.
multiplexing and there's multiplexing.
Yeah,
Getting them all work together is different from having a lot of assays together and one working. So that is a key
Yeah.
point you make there, Alex, because
Yeah.
that's often mistaken.
Yeah, thank you so much for this very interesting debate. I'm sure like there will be a lot of questions that will come up from this discussion on the level of multiplexing, the number of targets that we can get in a single well. I think that's the future of where the next few years of digital PCR will come. So with that, I want to thank you very much, our speakers, and I want to thank the audience for attending this session today. So then with that, I'll close up the session.
Thank you.
Thank you very
Okay,
much.
thank you.
Bye.
Bye-bye.